Okay, hello everyone. Welcome back. So we're now going to just do our last video. It's going to be the second part of proteins, and I'm also going to discuss nucleic acids very briefly. We have a full section on nucleic acids coming up, so we're just going to touch on it briefly right now. So last we spoke was about proteins, and this is a continuation about proteins. We're going to be discussing enzymes. Enzymes are proteins that catalyze the reactions of life. Enzymes are essentially proteins with specific shapes that are going to catalyze. They're going to speed up the synthesis or digestion of molecules. They react with very specific substrates or reactants and produce end products. So here we have our substrate, which is hydrogen peroxide. We have our enzyme, which is catalase, and it's going to interact with hydrogen peroxide. Our substrate and enzyme are going to interact, and it's going to form these end products. It will form two water molecules and oxygen gas. These are our end products. So the enzyme catalase reacts with hydrogen peroxide to produce water and oxygen. So enzymes can be affected by many factors. This includes increased temperature as well as a change in pH, which can make a protein or an enzyme denature. So in the top, we show a normal protein. When it's exposed to either an acidic environment or high temperature, it could denature and it will flatten out. So I spoke about this earlier with curly hair when you go get a perm, how those disulfide bonds are going to break. So it's going to break those bonds that create that secondary structure in hair, and it's going to cause it to become denatured and flatten out. So usually denatured proteins cannot bind up again. However, there are some exceptions. So if you think about your food, when you cook your food, you expose it to high temperature, your food can't go back to how it was previously. So after it's denatured, it can't, I guess, renature. But when you think about hair, it is able to build those bonds again. And here is shown with an enzyme. So you have your enzyme, you have your substrate. When you expose your enzyme to high heat, it can cause the enzyme to change in shape so that the active site is no longer able to fit into the substrate. And in all honesty, this is not my favorite depiction of denaturation because this is a lot of times people talk about enzymes and substrates as this key and lock method. So essentially your enzyme is your lock, or your basically this is your key and lock combination or complex, and they both have to fit in perfectly to be able to interact. However, that's not always true. Your enzyme can actually have a different configuration from your substrate, and then when your substrate binds, your enzyme can clench the substrate. So this isn't the best depiction, but essentially all we're trying to say is that the enzyme can lose its properties when it is exposed to high heat or to differences in pHs, and it will cause it to no longer be able to bind that substrate. So I'm sorry, my dog is barking in the background. So enzymes are proteins with specific shapes that catalyze, speed up the synthesis or digestion of molecules. They react with specific substrates and produce end products. This is what we already kind of touched on that first slide. The enzyme catalase reacts with hydrogen peroxide and produces water and oxygen. So over here we have our catalase, which is our enzyme, and we have our substrate, which is hydrogen peroxide. So we have a free enzyme and substrate. When they come in contact with each other, they bind together, so they create this substrate enzyme binding, and a reaction occurs. So think of your catalase as an open hand, and then when the substrate comes into that catalase, that open hand closes, so it clenches that hydrogen peroxide, and a reaction is going to occur. So when it clenches this, there's a configuration that's going to happen to this hydrogen peroxide. So when the catalase, the enzyme lets go, you now have 2 hydrogen molecules and 1 oxygen. So when it lets go, you have the 2 hydrogen molecules, the oxygen, and your enzyme catalyzed, and this is the release of products. So if we were in class, you would actually do an experiment. This experiment's pretty cool that can demonstrate this reaction. So you have potatoes, you have the enzyme catalase found in potatoes, and we would use these potato cores to core the inside of potatoes, and we would use hydrogen peroxide to test this experiment. So essentially you would have 4 treatments. You would have an unaltered potato core, a microwaved potato core, and an acid soaking potato core, and a chopped potato core. And then you would measure the reaction rates after a set amount of time by measuring the height of bubbles created in the test tubes. And I'm going to walk you through this very slowly. So I'm kind of just speeding up here, but essentially you're going to have your potato. Your potato is going to be the source of the catalase enzyme. Your enzyme is catalase. So you're going to use those potato cores, and you're going to core a piece of the potato. You will cut that core into 4 equal pieces that will be 2 centimeters long in length. And then you will have 4 test tubes. And those 4 test tubes are going to have 4 mils of hydrogen peroxide in them. So you're going to have 4 potato pieces, 2 centimeters in length, and 4 test tubes with 4 mils of hydrogen peroxide. Your first potato core is going to be unaltered. So you're just going to essentially grab that. Let me see if there's a better picture here. Okay, here we go. So you're in your first test tube, you're going to have your 4 mils of hydrogen peroxide. Your potato core that's unaltered, you're going to drop that in and look at the reaction. Oh, I even have a better picture here. Sorry about that. So like I said, first test tube, 4 mils of hydrogen peroxide. That is your substrate. You have your 2 centimeters of your potato core. That is your source of catalase. And then you're going to measure the reaction. You're going to measure the end products, which are essentially 2 molecules of water and oxygen that is produced. Your second condition is your 4 mils of hydrogen peroxide. And your potato core, this potato core was put in the microwave for 2 minutes. Then once it was cooled, you put it in your test tube. And then you would measure the reaction. So remember, when your catalase or your enzyme is exposed to high temperatures, it is denatured. So here you see no enzymatic reaction because your enzyme is denatured. It's not functional. Your third condition would be your 4 mils of hydrogen peroxide. And here you're going to take your 2 centimeters of potato core and you're going to chop it up into smaller pieces. So here you're increasing the surface area and increasing the amount of enzyme you have. You have more catalase in this condition. You put that into your test tube and then you measure the reaction. And then your last condition is going to be soaking your potato core into an acidic solution. So you expose it to low pH, and this will denature your enzyme. So you expose it to this acidic solution for 10 minutes, and then you put it into your hydrogen peroxide, and you will measure the enzymatic reaction. So remember, when you expose your enzymes to different pH conditions, this will denature your enzyme. So now it is non-functional. So here are some videos you can watch this. There's different YouTube videos where they show this done, and you can watch these and answer the questions in your lab manual. And here are some questions to think about. So why might the purpose of an unaltered potato core treatment be? So this is essentially to establish your baseline or to be your control group. So which treatment, microwave chopped, acid soaked potatoes, do you believe would result in the greatest amount of bubbles? Why? So for question 2 and 3, you would have to make predictions. And then question 4 is, how might an acidic environment and an increase in temperature affect the enzyme catalase? So remember, you're exposing your enzyme to high heat or acidic environment, you are going to denature your enzyme, making it non-functional. So lastly, I'm just going to briefly talk about nucleic acids. So the monomer for nucleic acids are nucleotides. Nucleotides consist of a nitrogenous base. So this is shown here, our nitrogenous base, a pentose sugar, and a phosphate group. There are various examples of nucleic acids. You have DNA, RNA, ATP. So in DNA is composed of nucleotides. These are the monomers. And you have four nitrogenous bases. You have adenine, cytosine, thymine, and guanine. They are shown up here. DNA, the pentose sugar, is called deoxyribose, and DNA is double-stranded as shown here. RNA is composed of nucleotides as well. Here, the four nitrogenous bases are adenine, cytosine, uracil, and guanine. So RNA has uracil instead of thymine. The pentose sugar in RNA is called ribose, and RNA is single-stranded. So cytosine, uracil, and thymine are all considered pyrimidines, and guanine and adenine are considered purines. So purines have these two ring structures. They have these two rings. Pyrimidines only have one, and these connect with each other. So what you will see is that guanine is going to pair with cytosine. As shown here, guanine pairs with cytosine. When guanine pairs with cytosine, 3 hydrogen bonds are formed between these two nucleobases. And then when adenine pairs with thymine, so this is in DNA, 2 hydrogen bonds are formed between these two nucleobases. So for our purposes, that's all you have to know about nucleic acids up to this point. Make sure you guys still read your guys' lab manual for activity 5 and activity 6, because there are some details I left out here in this presentation that are beneficial to understand these concepts. So don't forget, your guys' quiz is due Sunday by 11:59 p.m. and let me know if you have any questions.